Again, thank you all very much for having me, and thank you for taking time out of your night to listen to me talk. This is something that I'm really excited about, and that I hope is going to be really revolutionary for our field over the coming years and coming decades. I have to admit, I was very excited when I got the email invitation to give this talk, and I got my hopes up. I was lucky enough to visit your country about a decade ago, midway through my residency, and it was really a trip of a lifetime. And I hope someday I get the opportunity to return, and look forward to COVID abating and all of us getting back to being free to travel the world and see all sorts of great stuff again. Tonight I'm going to be discussing xenotransplant, which probably many of you have seen in the news recently. I think it's a tremendously exciting time for the field of xenotransplant, and I think we're on the cusp of starting to move this technology into humans. I'm going to briefly touch on some of the history of the field just to give everyone a sense of perspective, then discuss the kind of major advances that have happened really over the past 7 years that have pushed this from being a dream to something that's on the cusp of reality. And then I'm going to want to review the recent experiences moving xenotransplant into human recipients in a fairly limited way, and discuss what I think the next steps are to realize this dream. I'll start the caveat that most of what I'm going to discuss draws from the literature on xenotransplant of kidneys and to some extent hearts. Much of the same as science applies to islets as well. I'm not going to talk much about xenotransplant of lungs or livers, because those are the organs that are farthest away from being a clinical reality in people. And if people have questions about that or want to talk about that some more at the end of the talk, we can kind of talk about why. Like much of the rest of the science in our field, xenotransplantation has been driven by the shortage of organs for transplant. This slide has up the metrics in the United States about how many organs we need compared to how many we have. But the story is similar more or less across the globe. There are more people in need of transplants than there are organs available. And the magnitude of this problem is vast. And it's so big, it's nearly impossible to completely bridge this gap without something truly revolutionary. And there's a lot of good technology and a lot of good innovation out there to try to get more donors, things like increasing living donation, more aggressively utilizing marginal organs, technologies like ex vivo perfusion. And I think all of those have the potential to partially fill the gap, but truly solving this and making sure there's an organ for everybody in need requires a massive supply of new organs. And that's, I think, where xenotransplant comes in. The idea of using animal organs to bridge this gap actually goes back quite far. In modern transplantation, clinical xenotransplants start in the 1960s when Tulane University performed a series of 13 kidney xenotransplants over a time period of about 2 years. At the time these were conducted, there was not widespread use of chronic dialysis for patients with end-stage renal disease. And there was limited availability of deceased donor kidneys, as well as a fair bit of reluctance to pursue live donor kidney donation. So the Tulane group turned to chimpanzees as donors for renal xenografts. They transplanted these into 13 human recipients, many of whom did quite poorly, but actually one of whom did relatively well and went on to survive for 6 months with a functioning chimpanzee kidney. Around the same time, Tom Starzl published his own case series of 6 patients who underwent xenotransplants from baboon donors, and he achieved a maximum graft survival of only 60 days. He looked at all of the grafts when they failed and found that there was a heavy cellular infiltrate and extensive tubular necrosis on the explant of these kidneys. And it led him to claim that chronic survival after baboon to man transplantation seems to be a virtual impossibility. Interest in xenotransplantation continued to grow through the 70s and 80s, again, primarily using primates as donors. Public interest and outcry kind of reached a peak in 1984 when there was a high-profile case of heart xenotransplant into an infant at Loma Linda University. The patient became known as Baby Faye and was born with hypoplastic left heart syndrome. And she received a baboon heart transplant done by Leonard Bailey and lived just under a month after that transplant. The attempt got a lot of news and led to a sharp division in the medical community regarding the ethics of xenotransplantation in light of what was considered to be a quite poor outcome for this infant, and the need for more studies before more human attempts at xenotransplantation were made. The limited success of these early primate to human transplants, combined with the high expense of using primates and equally importantly, the limited availability of primates, caused a reevaluation in the xenotransplant world and really led to the consideration of other potential donor species. David Cooper, who's now at Mass General, was one of the earliest proponents of the current thinking in the field, which is that pigs are probably the ideal animal donor. He based this view on the similar structure and function of pig kidneys to human ones, the ready availability and rapid development of swine, and the absence of major potential infectious organisms in pigs. Despite all of these reasons to attempt pig to human xenotransplant, the earliest attempts at transplanting wild-type pig kidneys into non-human primates demonstrated rapid decline of the renal xenografts, and pathology of these explanted grafts showed hyperacute rejection. This formed the basis for the first major discovery that has paved the way to where we are today, which is that these hyperacute rejection episodes were traced to preformed antibodies in non-human primates against major naturally expressed antigens on pig cell surfaces. And the most significance of these preformed antibodies was identified as being against the glycoprotein GAL, with antibodies to two other glycoproteins, NuGC and SDA, also playing a significant role in hyperacute rejection of xenografts. This slide just shows that concept graphically. Because the majority of pig cells express the GAL antigen, and the GAL antigen is not found in humates or other primates, we've preformed antibodies against this protein, similar to what many humans have against a ABO blood antigen mismatch. When a pig organ is then transplanted into a human or primate, these preformed antibodies bind GAL, activate the complement cascade, and result in hyperacute rejection, typically within hours, causing graft failure. And again, this is just like we see when if you see ABO incompatible transplant in humans. The first major advance to overcoming this came with the development of a GAL total knockout pig in 2003. And grafts from these pigs were largely able to avoid hyperacute rejection when transplanted into non-human primates. But they failed to achieve long-term survival. These experiments were conducted with the genetic GAL silencing, induction immunotherapy with thymoglobulin, and triple maintenance immunosuppression with steroids, mycophenolate, and tacrolimus. But most groups using these donor pigs only report survival in the 5 to 20-day range. And failure of these grafts was largely associated with a marked increase in pig-specific antibodies following the transplant. These early failures with the GAL knockout animals led many in the field to conclude that additional genetic modifications are necessary to blunt the xenoimmune response to pig organs. And to achieve this goal, several groups began developing genetically modified pigs that, rather than having things knocked out, had human transgenes that were designed to provide some protection against the antibody-mediated immune response and the resulting complement activation. The attempts at these transgenic pigs actually date back to 1995 when Dr. White in Cambridge created the first transgenic pig that contained a human decay accelerating factor gene. which slows the complement cascade after antibody binding. This concept has subsequently been expanded with efforts to introduce other transgenes involved in complement and coagulation regulation, also attempting to reduce the damage done by antibody-mediated rejection. And the addition of these transgenes, particularly human decay accelerating factor, does help decrease C3 deposition and reduce antibody-mediated rejection severity in xenotransplantation. These single transgene animals again proved insufficient to deliver true long-term survival, with most groups reporting a pig to primate renal xenograft survival of around 55 days when using a porcine graft with just the human DAF transgene. So these early attempts clearly are not laying the foundation for what we've seen over the last 6 months. And the field really hit an inflection point in 2015 when survival in preclinical models started significantly increasing. And this is a graph that only goes through 2018, but the trend has continued into the 2020s. These improved outcomes are what have paved the way for the recent steps into moving xenotransplant into humans. And I want to take a few minutes to review the advances that have led to these dramatic improvements in survival. And there are multiple advances, there's no single cause. And I think these advances together are additive. And I want to focus on 3 specific advances that I view as being particularly key. One being the increased ease of genetic editing, 2 being improved xeno cross-matching, and 3 being improved immunosuppression for the xenotransplant recipients. The first of these major increases in xenograft survival came in 2015 when Owasi et al. Reported survival of 136 days after xenotransplant, again using a kidney xenotransplant. They used a donor animal that was a total gal knockout, and then it had a total of 5 transgenes, including 2 for human complement regulation and 3 for human coagulation regulation. And obviously, this magnitude of genetic editing is a major step forward compared to the single knockout or single transgene animals that were being studied in the early 2000s. In addition to this more extensively edited pig, this group employed a more aggressive immunosuppressive regimen that included both B-cell depletion and co-stimulation blockade. I want to first focus on the genetic modifications in the pig, because the change from using single gene-edited animals to multiple gene-edited animals is really revolutionary. And this more extensive genetic engineering has really only been unlocked in the last decade or so, as technologies like CRISPR-Cas9, TALEN, and zinc finger nucleases have dramatically reduced the barrier to targeting multiple genes in a single animal. This chart includes some of the genes that have been targeted in various preclinical xenotransplant models. It's not exhaustive, but I think it contains many of the more compelling and common gene edits that people have investigated. And I want to briefly go through a couple of them to give you all a sense of what's being considered. The first column is probably the most important column, and it contains the glycan knockouts. These are generally accepted as the most critical genetic edits because of the extent of preformed antibody in primates against these glycan antigens. Again, like I said earlier, GAL is the most dominant of these and has been the most extensively studied in non-human primates. And it's also the dominant source of preformed antibody in humans. NU5C and SDA are also major antigens and major sources of preformed antibody in humans, but they've less extensively been studied in the preclinical models in non-human primates for a couple reasons. One being that SDA occurs naturally in old world monkeys, so knocking it out has no advantage in experiments using those recipients. And also that creating a triple knockout of all 3 of those glycans exposes a new target glycan epitope that most primates have preformed antibodies against, but luckily humans do not. So a triple knockout animal, we think, is posed to do worse in non-human primate models than it would perform in a human recipient. And therefore, despite the relative lack of complete triple glycan knockout data, those animals, I think, are viewed by many as probably the single best foundation for human xenotransplants. With that, there is also, I think, despite the improvement in evasion of preformed antibodies with these triple glycan knockout animals, there are still antibodies against minor antigens. And I think most in the field continue to believe that there is advantage to continuing to use human complement and coagulation regulatory proteins as transgenes to reduce any residual damage and to blunt antibody-mediated processes in the xenograft. Another number of other anti-inflammatory proteins, including hemoxygenase 1, CD47, and A20, have been explored as well as a couple of immune regulatory molecules, all aiming to reduce the risk of either antibody-mediated or cellular rejection in these xenografts. And then in the last column there, which I've listed there are a couple of miscellaneous edits that may be important when we think about non-immunologic consequences of xenotransplantation. Among the most important of these are knocking out human growth hormone receptor, because in some species of non-human primates, we've seen massive hypertrophy of xenografts, which is a big problem in cardiac xenotransplants, where if the heart hypertrophies too much, it essentially runs out of space in the chest. But can also be a problem in kidney xenotransplant. If the kidneys massively hypertrophy, they start to cause problems with the domain of the abdomen. There's also been some CRISPR-Cas9 technology that's looked at inactivating something called porcine endogenous retroviruses, which are a class of virus integrated into pig DNA that theoretically pose a risk of infection and malignancy in humans. And I think more than actual demonstrated risk, they have been a big concern for a number of the regulatory bodies. So the ability to use genetic editing to inactivate these is exciting because it potentially lowers the regulatory hurdles to moving this technology into people. Before I move on from the slide, I also want to point out that it's not just the choice of transgenes that matters in these animals. These transgenes can be put on a number of different promoters, each of which changes the expression levels of the transgenes and potentially alters counter-regulatory mechanisms. So when we think about what sort of genetic makeup we want for a donor animal, we have to be thinking both about the genes themselves and the level of expression we want of these genes. So there are a tremendous number of variables involved in designing these genetically edited animals. Beyond just this more sophisticated gene editing, the other major advances that we've seen in preclinical xenotransplant survival come from improved understanding of xenoimmunology. And I want to start with our improvements in avoiding antibody-mediated rejection, which probably remains the most vexing immunologic issue in xenotransplant. Many of the improvements we've seen over the last 7 years actually come from applying what's really quite old technology, which is the cross-match, and trying to make better decisions about which primates are potential xenotransplant recipients. The earlier experiments in xenotransplant treated all animals in a recipient species as having the same fundamental immunologic risk after a xenotransplant, rather than taking this individualized approach with the cross-match that we use in humans. And this assumption was driven largely by the dominance of anti-glycan antibodies, particularly those against gal. But in the last decade, we began to challenge this assumption. And it was really as gal knockout and transgenic donor pigs were more widely used in preclinical work, we began to observe that there was a lot of variability within individual groups within an experiment. And we'd see that most xenografts would survive a few weeks, some would be rejected faster, but some would make it to almost 100 days. And this heterogeneity in outcomes led many in the field to question whether the preformed antibodies against minor non-gal antigens were responsible for this accelerated rejection, but not all animals, and whether the titers of these antibodies might explain these inconsistent experimental results. In this hypothesis, there would be a spectrum of titers of antibodies against minor antigens within a given species. And a recipient with high titers of these antibodies would still have antibody binding and rejection despite transgenes and despite glycan knockouts. But recipients with lower titers, the transgenes would be sufficient to protect them from antibody-mediated rejection in these low titers. This hypothesis was most thoroughly tested at Emory, where they screened 33 rhesus monkeys for preformed antibody against donor cells from gal knockouts with the human decay accelerating factor transgene using flow cytometry. And you can see the distribution of antibody titers here. And you can see that there was a wide range of titer within those 33 rhesus monkeys. They then performed xenotransplants in these animals, choosing the animals at each extreme, the highest and lowest antibody titers for their experiment. The results were both striking and unsurprising. Recipients with low antibody titer survived significantly longer than those with high antibody titer, with a median survival of 160 days compared to just 6 days in the high titer animals. At rejection, xenografts from the high titer monkeys showed characteristic findings of AMR with interstitial hemorrhage, edema, and antibody deposition. These results, antibody-mediated rejection in animals with high antibody titers, are expected, but observing it despite the use of gal knockouts established that xenoreactive antibodies against minor non-gal antigens play a huge role in the anti-xenograft rejection. And as importantly, this experiment demonstrated the profound impact that simply screening monkeys for these preformed antibodies could have on survival, with one recipient making it all the way to 310 days with no further genetic modifications of the donor animal and no other adjustments to immunosuppression. We see a similar phenomenon in humans, and this is a study from Indiana and Emory, where the sera of 84 human recipients was exposed to pig PBMCs from either double or triple glycan knockout pigs. Similar to what we saw in the rhesus monkeys, you see a variable distribution of preformed antibody titers to the double knockout pig cells in the human subjects, and to a lesser extent, continued variability in titers against the triple knockout pig cells. Notably, by the time we get to triple knockout animals, a full 90% of human recipients fall into the bottom left corner of graph B, which indicates a likely negative cytotoxic crossmatch. But this finding reinforces the need for good cytotoxic crossmatches as we move xenotransplant into human clinical trials to make sure that we're choosing people who are not at risk for accelerated rejection. The crossmatch may be particularly important in allo-sensitized human candidates because there's additional cross-reactivity between some anti-HLA antibodies with swine leukocyte antigen epitopes, particularly in class 1. This finding, again, has significant implications for the design of the initial human clinical trials, because a subset of patients who have otherwise been discussed to be a particularly attractive trial enrollee, which is the highly sensitized patients who have decreased access to human transplants, may pose additional challenges in the xenotransplant field. And using them in the initial trials may predispose these trials towards failure. The recognition of the importance of preformed antibodies is not just limited to the use of the crossmatch. It's also informed the addition of rituximab or other B-cell depleting agents to many xenotransplant immunosuppressive regimens. The survival curve shown here, which comes from an NIH and University of Maryland study on cardiac xenograft survival, demonstrates a fairly dramatic increase in graft longevity with simply the addition of B-cell depletion to an otherwise relatively standard immunosuppressive regimen. There have also been some important advances in xeno immunosuppression focused on T cells, most notably through the application of co-stimulation blockade. There are a number of pathways involved in co-stimulation, but in human allotransplant, only the CTLA-4 pathway is commonly targeted with therapeutics, namely belatacept. But in preclinical models of xenotransplant, we've had the opportunity to directly compare blockade of this and other co-stimulation pathways, and it's uncovered some somewhat surprising results. In this pilot study, which again comes from the Emory Xenotransplant group, they compared recipients of xenografts treated with the CD40L inhibitor 5C8 with those treated with either anti-CD28 antibody or belatacept. And they found that there was markedly enhanced survival in the 5C8 treated animals. This finding has been replicated in another study from Indiana shown on the right, and has really reinforced a new understanding of the importance of the CD40, CD154 co-stimulation pathway in rejecting xenotransplants. And conversely with that, the importance of blockading this pathway to avoid rejection. Blockade of this pathway has been widely adopted in many subsequent preclinical experiments. Unfortunately, the importance of this pathway also creates a little bit of a conundrum in terms of designing human clinical trials, because there are no FDA-approved CD40, CD154 agents for human use right now. 5C8, the agent used in the Emory and Indiana trials, and probably the most widely used agent in other preclinical xenotransplant experiments, was in phase 2 trials, but was withdrawn because of a high incidence of thromboembolic events. There are currently about a half dozen other CD40, CD154 blockade agents that are in either phase 1 or phase 2 trials. But the absence of an approved drug that acts on this pathway, I think, is going to create significant regulatory hurdles, at least in the US, where our regulatory bodies are fairly resistant to testing both a new drug and a novel gene-edited xenograft in the same trial. I want to end this section with just a brief summary of what I view as kind of the key lessons from the preclinical experience with pig to primate xenotransplants. First, I think from a gene editing standpoint, you definitely need a gal knockout animal. And in humans, I think additional knockout of NU5C and SDA is probably also extremely important, just because of the magnitude of the preformed antibody component to them. But I'll readily admit that that's been hard to establish in our preclinical models. I think there's also a role for human complement and coagulation regulatory transgenes to protect xenograft from antibody against minor xenoantigens. But I'm not sure there's been enough direct comparison of the various choices or their promoters to establish a really optimal regimen. I think the second lesson is that cross-matching is key, in that not every monkey and not every human, when we get there, are going to have an immune environment that can tolerate a xenograft. And success in clinical xenotransplantation in humans is going to demand that we identify which people are likely to do well and are at lower risk for rejection. And third, I think immunosuppression has to be more profound than what we typically use in human allotransplantation. At a minimum, it requires steroids, thymoglobulin, anti-CD20 B cell depletion, and co-stimulation blockade, preferably on the CD40, CD154 pathway. Induction with these agents creates significant immunosuppression. And I'm not sure we have yet a firm answer on what sort of maintenance regimen xenotransplant recipients will require long-term. And that's partially because there tend to be a lot of side effects of tacrolimus and mycophenolate in monkeys. Tacrolimus tends to cause pretty severe loss of appetite, and we end up holding it often in our monkeys. And similarly, the primates hate the taste of the mycophenolate. So it's difficult to figure out which one of them how much they're actually getting and how often they're actually taking it. Because if you glance away for a second, they try to throw it away. So between the profound immunosuppression we induce at induction and some of these issues with the maintenance agents, I'm not sure we have a firm answer for what sort of maintenance immunosuppression humans will require long-term with the xenograft. But I think to start, probably steroids, tacrolimus, and mycophenolate is a reasonable regimen, provided that co-stimulation blockade and T and B cell depletion is provided up front. I'm going to change gears a little bit here and focus on really the excitement of the last 6 months, which has been the human application of xenotransplants. Many of these experiments hit the mainstream press before they've hit the scientific press. But there have been several huge leaps forward, taking organs out of pigs and putting them into people. I want to go through what we know about these efforts, although again, I'll caution you that some of this data is gleaned from mainstream press, some from rumors or discussions with collaborators, and some of what I'm going to tell you about has not yet been reported in peer-reviewed literature. So the first report of a xenotransplant from a pig into a human came out of New York University last December, when a team there transplanted a pig kidney into a brain-dead human. NYU has subsequently repeated the experiment in another brain-dead human, and they've not yet published their data. The organ used in this experiment actually came from a less extensively genetically engineered pig than the ones we've been discussing. It had only a single gene GAL knockout, and it comes from an established commercial pig line called GalSafe, made by Revivicor. And this actually is not a cloned pig. That fact has some significance because cloned animals tend to be quite unhealthy due to some of the epigenetic dysregulation that accompanies the cloning process. And we've often observed that cloned animals have infectious issues and can have anatomic abnormalities, duplicate renal veins, renal arteries coming off of external iliacs, and they can have trouble with weight gain and other issues. Most of the more extensively gene-edited pigs are only available as clones. And that's partially because of reproductive issues that accompany making multiple gene edits in these pigs. And it's also partially just because in some cases there's not been adequate time to establish a stable animal line. But despite this being a less extensively gene-edited animal, there is some potential advantage to using a non-cloned animal, which I suspect is why they chose this line for this experiment. I've not yet seen a report of what cross-matching strategy, if any, they used in this experiment. And most of the details of the immunosuppressive regimen have not yet been published. But in the lay press, they did report that the transplants also included a pig-to-human thymus transplant, presumably to generate some degree of chimerism intolerance. Follow-up in these experiments was about 2.5 days after transplant, 54 hours. And the kidneys were reported to have made urine and cleared creatinine. Again, I don't know whether the native kidneys were removed, whether both of these donors had pre-existing renal failure, or what kind of strategy was used to distinguish renal function in the transplanted xenograft compared to native kidneys. And the authors reported that there was no rejection, although, again, we have not yet seen publications showing the biopsies or photos of the kidneys. I think overall this is a really exciting conceptual experiment and a good indication that we can avoid hyperacute rejection. But there obviously are a lot of questions about this experiment that I hope will be answered in the coming months as they publish the results. A more completely reported and conceptually similar experiment was conducted at the University of Alabama at Birmingham at about the same time. And this was reported in the popular press and the scientific press at the same time. In this case, they reported transplanting 2 pig kidneys into a brain-dead human recipient and at the same time performing a bilateral native nephrectomy of the recipient's kidneys. The experiment was sponsored by the same company as the NYU experiment, Revivicor, but in this case, they used a more extensively gene-edited donor pig, which had 10 total gene edits, and it was a cloned animal. The 10 gene edits included the targeted insertion of 2 human complement inhibitor genes, which were decay accelerating factor and CD46, transgenes for 2 human anticoagulant proteins, TBM and EPCR, and then transgenes for 2 immunomodulatory genes, CD47 and HO1. And then they deleted all 3 of the major carbohydrate antigens, and they also deleted pig growth hormone receptor. The UAB group performed a flow cytometric cross-match pre-transplant to make sure there was not prohibitive levels of anti-pig antigen and antibody in the recipient. And their immunosuppressive regimen consisted of a methylprednisone bolus, anti-thymocyte globulin at a dose of 6 milligrams per kilogram, anti-CD20, MMF, and tacrolimus. They notably did not use co-stimulation blockade. And they followed the recipient for 77 hours until there was prohibitive hemodynamic instability from brain death, and at which case they ended the experiment. These are the results of their flow cytometric cross-match, which again show relatively low levels of preformed anti-pig antibody in this recipient. And here are their functional results, which is I think where there are a couple problems with this experiment. They left stents in each of their ureters so they could distinguish urine production from each kidney. And they put the right kidney in first and then the left kidney. The right kidney, which again is the one that went in first and is shown in green, made urine immediately, although the magnitude of urine production tapered off as the experiment progressed. The left kidney, which went second, made pretty minimal urine, except for a couple hours midway through postoperative day 1. And in the bottom, you can see that the recipient's creatinine and BUN continued to climb even after reperfusion of the xenograft kidneys. So functionally, some urine production, but not a great creatinine clearance in these experiments. The authors performed serial biopsies that I think partially explain the relative lack of biochemical function. The biopsies showed multiple fibrin thrombi in the glomeruluses, and they also showed diffuse glomerular capillary congestion with swollen endothelial cells that was consistent with thrombotic microangiopathy. And this was evident by 16 hours on post-op day 1. With this TMA, they saw progressive tubular injury, extensive acute tubular necrosis, but CD4 staining, C4D staining was negative throughout the experiment, which I think is a good sign that there wasn't a major component of antibody-mediated rejection. I still think this is an exciting experiment, and that there are a number of lessons to be learned, also a number of questions that this raises. One is this was a really strong logistical feat to pull all this together, and I think it was very well conducted and very thorough in terms of the data they collected from these animals. And it showed that we can avoid hyperacute rejection using this regimen of this animal. And in the short term, at least, I didn't show this data, but they showed that there was no evidence of porcine endogenous retrovirus transmission into the recipient, which again, I think is important primarily from a regulatory standpoint. The questions that come up from this experiment mainly stem from the poor urine output and the lack of creatinine clearance. And I think the question is, was this due to instability and the brain death milieu of the recipient? Or does this, the TMA and the lack of function, point to deeper issues, either with the selection of genetic edits of this particular donor, or perhaps the immunosuppressive regimen and the absence of co-stimulation blockade? And I think we have to think about how we avoid TMA in future experiments. And this really is different from what we've seen in non-human primate experiments using kind of similar gene profile donors in similar immunosuppressive regimens. And I think we need this to see more repetitions of this experiment to try to distinguish whether this was really a recipient brain death issue or whether this was some more fundamental issue with the kidneys. I think the other question it raises is whether these kidneys are very sensitive to cold ischemic time. In most of the non-human primate models, the groups managed to keep their ischemic times relatively short. But I think the difference in urine production between the kidney that was reperfused first and the one that was reperfused later may indicate that these kidneys are more sensitive to temperature fluctuations and the time out of body. The most successful human xenotransplant is kind of next up. And it's also the only one that went into a living human. This was a cardiac xenotransplant that was performed on a 57-year-old male with end-stage heart failure who was deemed not to be a human heart transplant candidate at the University of Maryland. It occurred not as part of a clinical trial, but under a compassionate use agreement. The recipient received a heart from a 10 gene-edited pig made by Revivicor. I think it's likely that this had the same gene profile as the UAB experiment, although I haven't seen the actual gene profile of this donor published. And again, the cross-matching strategy is unknown. The full immunosuppressive regimen has not been explained yet, but they did explain that it included an experimental drug made by Kaniska, which is a biotech company in the States. And just by browsing their website and their products in the pipeline, I suspect this is an anti-CD40 monoclonal antibody. So acting on that CD40, CD154 co-stimulation pathway that they currently have in phase 2 clinical trials for rheumatoid arthritis. The recipient did require ECMO coming out of the OR immediately after the xenotransplant, but came off ECMO and survived for just under 60 days before passing away. And the cause of death has not yet been reported. I think there are a number of possibilities considering that he was someone who was deemed to be too sick to be a candidate for a human transplant. It may have nothing to do with the xenotransplant, or it could have something to do with either an infection related to extensive immunosuppression, rejection, or something else attributable to the xenotransplant. I'm going to end with just a couple minutes on what I think the next steps are. And I think these recent experiences have brought a lot of energy and excitement to xenotransplant. And they naturally raise the question of whether this technology is ready for primetime human use. And in reality, I think we're already seeing the beginning of clinical trials in xenotransplant, at least in xenotransplant type products, internationally. In China, there are a couple of groups that are trialing corneal xenotransplants. And the first clinical trial of eyelid xenotransplant under a national regulatory oversight is currently underway in New Zealand, although that doesn't use the degree of immunosuppression or gene editing we've been discussing for renal or heart xenotransplants. In the US, the pathway forward is more complicated, and it requires coordination between 3 regulatory bodies. The Health Resources Service Administration has oversight over transplant of vascularized human organs, and the FDA and the FDA Center for Veterinary Medicine regulate xenotransplant products and genetically engineered products, respectively. But hopefully the recent NYU, UAB, and Maryland experience signal growing enthusiasm from the regulatory bodies to trying to help us find a path to move this technology forward. The FDA has put out a statement that initial efforts in xenotransplantation should be limited to patients with serious or life-threatening diseases for whom adequately safe and effective alternative therapies are not available. I think many of our patients on the wait list meet this threshold, so I think we should be able to move this technology towards clinical trials. The International Xenotransplantation Association has also put out a consensus statement with kind of similar criteria, but also includes those who are disadvantaged in the current organ allocation schemes. Um, I think one of the most crucial decisions as we move forward is which patients we are going to include in the first clinical trials. And it's really a question of which patients we define as the most disadvantaged under current conditions and which ones we think are likely to do the best with xenografts. I think we need to think about setting these trials up for success. And I think that probably means that we have to avoid allosensitized patients and those who have rapidly recurrent kidney diseases after prior transplants in the first trials. And I'd like to instead see us focus on people who don't have living donors and who have limited wait list time and therefore are looking at prolonged periods of dialysis before they have access to a human allo transplant. I think those patients are the ones who are likely to do the best with a xenotransplant. There's also a question of whether hearts or kidneys is going to make it to a real formal large human clinical trial first. As an abdominal transplant surgeon, I'm obviously biased here and think kidneys should go first because I think the consequences of an early graft failure are more easily mitigated with transplant of a kidney, where we can simply do a transplant nephrectomy and return a patient to dialysis. I think it's less high stakes, and I think that these initial clinical trials are going to teach us a lot about the immunology of these grafts in humans. And I think that it makes sense to collect that data in a scenario where failures are not going to lead to patients dying. Scientifically, as we move forward, I think we have a couple of big challenges. And I think one of the big ones is we need to define the optimal sets of gene edits for these organs and the best immunosuppressive regimens. Various groups have tested a lot of gene edits, and I don't think we've come up with a comprehensive way to rank their relative value and decide which ones are really critical. I think likewise, we've made a lot of progress towards defining the best immunosuppressive regimen. And I think it has to be, again, a fairly aggressive regimen that includes TMB cell depletion and co-stimulation blockade. And we have to make sure that humans can tolerate that regimen. In monkeys on these heavy immunosuppressive regimens, we do see a lot of things like parvovirus infections and other opportunistic infections like CMV. But obviously monkeys are housed in less sterile conditions and less ideal conditions than humans. So hopefully the opportunistic infections will prove to be less of a challenge in humans. I think we also need to work on standardizing cross-matching techniques. And I hope we can move towards single antigen Luminex beads like we're using for virtual cross-matches in humans, which also would give us access to rapid diagnostics for antibody-mediated rejection and detection of DSA after a transplant. Another exciting avenue for the future, I think, may be personalized gene editing of these grafts, particularly editing SLA epitopes to match recipient HLAs. And then, as I alluded to at the beginning, I think there's a lot of work that's needed to take this technology that is nearing human use in kidneys and hearts, and also make it work in liver and lung transplants. With that, I'm going to wrap it up. I want to thank a couple of people here on our Xenotransplant team at the University of Wisconsin. Dixon Kaufman and Tony D'Alessandro have been very instrumental in getting our program up and running here. And Stephen Kim, who's one of our fellows who experienced in xenotransplant from his residency, gave me a lot of help with this talk. And he is headed back to Emory next year to help with their xenotransplant program. And we look forward to building a stronger collaboration with the xenotransplant group there on his return.